with all right, we are now recording. Welcome everyone to Office Hours with Dave and Anita. This is episode 21 and we'll be featuring Luis Diaz Garcia. He's an assistant professor in viticulture and enology, and today's topic is why do we need new grape varieties? So just a couple of housekeeping items. Please make sure that you are muted during the program, and the mute button is usually on the bottom left of your Zoom window. So just make sure that you're muted, because I'm sure you don't want us to hear any side conversations or phone conversations that you may be having in the background. If you'd like to ask a question, there's a few ways you can do that. You can raise your hand with the Zoom reactions button, sort of over on the bottom right-hand part of your window. Or if you want one of us to ask the question, you can put it in the chat, or you can simply unmute and ask your question during a lull in the conversation or proceedings. We are very grateful to our extension and industry relations partners for their support. Without them, we would not be able to bring all of the extension programs that we do throughout the year to our stakeholders. And if you or the company you are working for are interested in sponsoring an office hours episode, please email Caroline or myself. It would really help us to continue to be able to provide these programs to you guys, our industry stakeholders, free of charge. I would also like to thank Caroline for helping with registration and helping to get the word out about this program. Of course, Dave and Anita for being here every time we do one of these, and also to Luis for his willingness to share his expertise today. And of course, all of you for joining us today. So without any more from me, I'm gonna throw it over to David Block, and he is going to introduce Luis today.
Great. Thanks. I should mention, because she didn't, that you were just listening to Karen Block, the Director of Industry Relations here, and the other person that helped us to organize this program. So thank you to Karen as well. So it's my pleasure to welcome you after kind of a little bit of a break from our office hours with Dave and Anita programs, back for a really great program today featuring our newest faculty member, Luis Diaz Garcia. Luis got his undergraduate degree in agronomy from the Universidad Autónoma Chapingo in Mexico. And then after getting his master's degree in plant biotechnology, also in Mexico, he went to get his PhD at the University of Wisconsin-Madison, where he worked on breeding cranberries. After he finished his PhD on cranberries, he went back to Aguascalientes in Mexico to a government research facility where he continued to do plant breeding, this time on guava and maize and various kinds of peppers, before he accepted the position here and started not quite a year ago, a little less than a year ago, here at UC Davis. We're very, very happy to have him joining the department, where he has become our grape breeder. Many of you will have known Andy Walker, who retired over a year ago. And to continue those efforts and have new efforts in grape breeding, we hired Luis. And so we'd like to welcome Luis to Office Hours with Dave and Anita and give him 15 minutes or so to talk about grape breeding and what he sees as the need for new grape varieties. And then we'll give everybody a chance to ask questions like Karen said. So feel free, as you're listening to Luis, to put questions in the chat or write them down for yourself, and we'll give you time to ask them, plenty of time to ask them a little bit later in the program. So with that, I'll turn it over to Luis.
Thank you, Dave. Can you hear me well?
Yes.
Okay, perfect. Okay, well, thanks so much for the introduction, and thank you, Karen, and all the ones involved in organizing this seminar. This is my first time in office hours with Dave and Anita, and I'm very excited about it. I was asked to talk about the reasons of why we need more grape varieties. And I know this is a very polemic topic. I think it needs a lot of discussion because there are many different angles. Some people think that we need more. Some people think that we don't need them, that we have plenty already. And I think there are very good reasons, and all of them are very valid and strong behind each idea, right? We need more, or we don't need them. So I was thinking about how to start this discussion, how to set up the stage to talk about this topic in particular. And I was thinking the other day about, what if I start with a counter argument, which is why don't we need new grape varieties? Because if you think about it, most of what we see in the market, in the wine industry, the consumer loyalty for the traditional varieties, all of that indicates that we don't need more cultivars. So let's start with that assumption. So in the world, almost in every country you can imagine, people grow grapes. So these can be native grapes that have been there for forever, or this can be grapes that were introduced at some point, but now they just grow well. This can be vinifera species, or regular grapes. This can be wild species, non-vinifera species. And even some people make wine out of these non-vinifera species. For example, in Mexico, where I am from, in the southern states, some people make wine out of Vitis stilifolia, which is a wild species. People drink it. It's not the best wine, but that's how it is, and that's perfectly okay. So all these grapes are very well adapted to these different conditions. It can be very hot climates, cold climates, different soil types, different pests and diseases. And so we have a huge amount of material, genetic diversity, phenotypic diversity, and that we need to explore in more detail. On top of that, we can expand this phenotypic variability through the winemaking process. So winemakers take all the grapes we harvest, and they can produce this amazing range of flavors and aromas. So this is a network with the most famous wine grape cultivars. So these are all the purple or white dots. And in green, we have all the most characteristic or iconic flavors associated with those particular grapes. So there are two clouds, one cloud in the top, which are mostly red wines. So they have a very unique characteristics in terms of flavor. And in the bottom, we have all the white wines, which also have a very unique profile. So we zoom in, we see all these very amazing, very diverse number of flavors, things like blueberry, dried cranberry, mocha, grilled plum, not only plum, but grilled plum. So the diversity is huge, right? So genetic diversity already we have available is pretty big. And on top of that, with the winemaking process, we can expand it. So even though we have such a great genetic diversity and cultivars all over the world, most of the grape production is based on just a few materials. So in California, we grow about 120 different cultivars, including wine grapes, table grapes, and raisins. And these two bar plots show the production in the state last year, 2022. As suspected, Chardonnay is by far the leading cultivar with almost 400,000 tons in the white grapes. In the red wines, Cabernet Sauvignon also produces almost the same amount. It's the most important cultivar. So the important message I want to give here is that about between 6 and 10 cultivars out of these 120 make up about 90% of the total production in the state. So if we consider the top 20, that's going to be about 99% of the production. And as a consequence, these important cultivars like Cabernet Sauvignon and Chardonnay, so these cultivars are planted across all 17 pricing districts in California. So for the ones that are not familiar, California has different regions, 17 in total. So basically these materials, because of their importance, the production, these are planted basically everywhere across the whole state. And not all the regions in the state are suitable to grow these particular grapes, right? Some places, like probably Napa or other places, are better, offer better climatic conditions and soil types to grow higher quality grapes. So what's the problem? So the problem is that climate change is going to affect the way we grow grapes. It's going to affect the way, the type of grapes we grow, and where we grow them. So this is a projection of what's going to happen with the suitability of grape growing regions around the world. So suitability is projected to decrease for some important wine grape regions, like in Europe, California, Chile, some places in Australia. And in some cases, it's going to be very drastic, like 50% is projected to be reduced in California in terms of suitability to grow grapes. Some other places at higher latitudes, like in Northern Europe or northern parts of North America, are expected. So these are places where we don't grow grapes at very high volumes right now, but in the future, these places might be favorable to do that. So climate change is also going to affect the spatial and temporal distribution of pests and diseases. And the problem is that most of the vinifera cultivars we grow lack the capability to deal with this type of stresses. So to grow most of these cultivars, what we have to do is we have management practices in the vineyard to, like fungicide application, things like that, to deal with all these challenges. And there are multiple problems associated with that. So we have to apply multiple times, and that represents a cost, and there's a negative impact for human health and the environment and so on. There is this issue with fungicide resistance. For viral diseases, for example, we mostly rely on prevention because there's not a natural genetic resistance on the cultivars we grow. However, non-vinifera vitis species are a good source of adaptive traits, and we haven't explored 100%. So I think there is a lot of potential here. And with the new molecular technologies we have today, we can develop new hybrid grapes with a very high percentage of vinifera, above 90–95% or even more. So that a small portion of the wild species that is used to develop these materials, so that portion carries only the adaptive traits, the genes that confer those resistance or tolerance to the different stresses we can find. And when I say molecular techniques, I'm not talking about GMOs or genetically engineered plants. So these are very traditional techniques. So we take pollen from one plant, we pollinate another plant that we think carry interesting traits that we want to integrate into new varieties. But now we just have better and more efficient methodologies or approaches to select these better plants, which makes the breeding process way more efficient. Wild species in particular are very useful to develop new rootstock materials. So right now, most of the global viticulture relies on less than 10 rootstocks. Most of them derive from just a few species, like the ones I'm showing here, Vitis riparia, Pests, and Perlandieri. But we have many wild species that we can use. Just in North America, we have about 20–25 different species, depending on the classification. And all of these species are very good sources of tolerance to nematodes, drought, salt. And some of them are easy to graft. So what we have to do is start combining all these traits into the next generation of materials. So do we need more cultivars or do we not need more? That's kind of the question. So this is what I think. So we have plenty of materials. That's a fact. But we need more data on these materials. And the problem is most of the data we have, at least for these minor and not very famous materials, is very anecdotal, right? And we know that for wine production, for example, the vintage is very important, right? The environment where these grapes are grown is very important, and it has a very strong impact. So what we maybe need is to have multi-year field evaluations. We need to evaluate wine quality more consistently so the data can be compared. But the problem is still that most of these vinifera-based materials still lack pest and disease resistance. So that's why we need to we'll rely on wild varieties species. So this hybrid grapes that we need to develop, I guess the first requirement is they have to be very high percentage of vinifera, above 99 or 95%. We have the potential to integrate multiple sources of resistance. So, for example, multiple genes that confer powdery mildew resistance or downy mildew resistance. There are different sources of these genes that have been identified in multiple species, Vitis piacenski, Vitis rotundifolia, murensis, and others. And it's important because having multiple stack genes or alleles allows to have durable and stable resistance, which is very important. We need to have different profiles for blending, including, for example, tenturier materials for color correction, stability, things like that. Wine quality is very tricky because, so what's quality, right? It's a very difficult trait to define. What we want to have maybe is not a very high quality hybrid grape. Maybe you just want to have an acceptable material that can be blended, that can be planted around the vineyard in those regions where there is a lot of pressure from diseases and pests. And I think there's no question that we need more rootstock materials. And I guess the limitations are, or the restrictions are less. Maybe we have more chances for these materials to be adopted. So what we need to do is combine drought tolerance, salt tolerance, nematode tolerance, virus tolerance into a single material. And I think we have the resources to do that. So I think we are almost 15 minutes. So this is what I have. I want to leave some time. I think this is a very important topic for discussion. So thank you so much.
Thanks. I think Anita has the first question for you.
Yes, sorry, I was unmuting myself. For some reason, I couldn't find the mute button after all these years. So this is an interesting question, but that was sent to us actually in advance. And so this is somebody that wanted to know if you can please cover new varieties for planting on slopes and suitable soil types. You know, if there's not enough things to worry about like drought and salt and disease and nematodes, there you go. I wonder if there's anybody that has actually worked on that to look at varieties that may be a better variety to plant in. I don't know, many times it's rocky soils on these.
I'm pretty sure there's people that know that. I'm not that person. I will leave that to a more experienced person, an extension specialist, or so.
Yeah, there isn't really any work on that, right? I don't think there's any kind of breeding happening that focuses on that. I'm just asking. You would know the research better than I do for breeding.
Yeah, well, we don't have that's not like a breeding target right now, and it has not been in the past. There are rootstocks and specific science that might be more favorable for to use in those kind of situations.
Yeah.
I'm not a viticulturist myself, so I don't want to give an incorrect answer to that. So
that was a trick question. I would think also root system would also have a huge influence on whether a specific rootstock is.
Yeah, definitely, because I guess those places, the soil depth is way lower, is smaller, right, than in flat terrains. So there's less space for the root system. So maybe you want to have a higher, bigger rootstock, things like that. But I guess it depends on a lot of factors.
So there's a question in the chat from Steve Ponzo. He's asking, why not use genetically modified organism material, GMO, using CRISPR or something like that? And maybe you can talk a little bit about what are the potential benefits and dangers of going that approach versus the approach that you talked about, that using more conventional breeding.
Yeah, so I don't know about the dangers. I don't think there are that many dangers. So there are two types of traits from the breeding perspective. We have single gene traits. So these are traits that are controlled by just one gene, like berry color, for example, or muscat aroma. So there's just one gene involved in that trait. So it's a gene with a very strong effect. And we have polygenic traits. So these are traits that are controlled by, it can be 100 or it can be 1,000 genes, but those genes have a very, very small effect. And for example, drought tolerance, yield, things like that. So in this case, CRISPR technology is going to work just for single gene traits because those traits have a very, very strong effect. And so why not? Yeah, so we can generate more variability, and that could be important. So at the end, in a breeding product, having variability is the first ingredient. So if you don't have variability in your breeding populations, there's nothing to do. I don't think we need it right now because we have plenty of diversity. Andy Walker and Olmo, for example, here collected thousands of materials all over the world. So we have different flavors, different aromas, different, almost like everything you can imagine, we have it. And or if we don't have it, it's somewhere else in USDA or it's in the wild, and we can collect it. So I don't think we need this type of methodologies right now because we have everything we need. But in the future, definitely that could be an important tool. It's also useful to validate gene function. So for example, part of my job is to identify, I don't know, what genes are involved in a specific trait, right? Because by doing that, I can develop molecular techniques to accelerate the screening process. That's how, for example, the genes that are related to berry color were discovered, right? Through genetic mapping. So CRISPR technology is a tool that can help researchers to, for example, knock out that gene and see if there is a change in the plant. And so from that perspective, this is a very useful technique. But sorry, yeah, go ahead.
I was going to say, just kind of following up on that. I mean, I guess to use CRISPR or something like that, and this is what you're saying, you need to know the function of genes, or which genes are providing or contributing to a function, whether it's a single gene or multiple gene. Can you kind of comment, like what percent of genes are understood, or what percent for all the important traits that you've talked about, are all the genes known, or some of the genes?
Yeah, well, yeah. There are, I don't know, in the lower end, there are about 30,000 genes in grape, right? And we know whether all these genes, what part of the chromosome. So we have a pretty good idea, and Dario Cantu in our department has doing a terrific job in that area. What these genes do and how they are regulated, that's a completely different story, and it's very difficult to do. So I guess it's very hard to say a number about the genes that we know, what are the functions. For many single gene traits, like Pests disease resistance, for example, so we have a pretty good idea about what could be the candidate genes involved. There are like 5 or 6. Berry color, the gene that controls if in what species of flower is going to be female or male. So we have a pretty good idea about that. There's still a lot of things to work on, and trying to understand how these different traits are regulated. So long way to go.
And can you comment? So, I mean, my understanding from talking to Andy over the years and other people is that, you know, it could be easily 15 to 30 years cycle of coming up with a new grape variety. And I'm wondering, you know, if you can talk about, you know, if that's still true and what you're doing to speed that up, and also comment on, and I think there's a comment in the chat that we'll get to in a second. Will using genetic engineering speed that up or not really?
Yeah, I don't know. So we have already, so for single gene traits, we use an approach called market acid selection. And we have been using that technique for, I don't know, 10, 20 years at least, different crops, including grapes. And it's a technique that allows us to predict what's going to be the phenotype of a plant before we actually see the phenotype itself. So for example, if we make a cross between a red cultivar and a white cultivar, the progeny maybe is going to segregate for that trait. So that means that half of the population of the progeny is going to produce white grapes, the other half is going to produce red grapes, but we have to wait, right? So 2 or 3 years until the plant develops flowers and so on and produce fruit. With market acid selection, we can, from the moment the plant germinates, little seedling, we can cut the piece of the leaf and we can make predictions about what the color of the fruit is going to be. So, for example, this was a technique used by Andy Walker to produce these Pests disease-resistant materials. So instead of waiting, I don't know, 5 years between generations, we can have a generation every 2 years. So this reduces very drastically the time required to produce a new cultivar. But still, we are talking about 10, 12 years from the beginning to the cultivar and release. Again, this only works for single gene traits. The problem is when we talk about the polygenic traits. So these are thousands of genes with very, very small effect. So the kind of approaches have to be different. And this is something that I want to focus in my program. There are all these set of tools, high throughput phenotyping approaches that are very allow us to screen way more plants compared with traditional methods. And at the end, plant breeding is just it's a game about numbers. So the more plants we evaluate, the more chances we have to identify the plant we're looking for. So this is what we are trying to do, right? So using market selection and combine it with novel phenomics methodologies, high throughput phenotyping methodologies to screen more plants.
We have someone who raised their hand. Dustin, do you want to unmute and ask?
Yeah. So Dustin Hooper, Sunridge Nurseries. I guess if you're looking at 20, varieties making up 99% of what is, generating revenue in the industry, why not focus on, studying how you could put the resistances into those varieties at a very high level and maybe figure out how to legally still call them the same thing. I think adapting to new varieties is probably the hardest thing, especially when those 20 varieties take up the bulk of, generating revenue. It's not like you're going to breed something and it's going to come in and take 5% of the revenue. It's going to take, decades for something like that to come up on acreage and things like that. But if you could breed a capsaw with resistance that has the same characteristics as a current capsaw, that would probably be more relevant to keeping the industry moving forward.
Yeah, thank you, Dustin. Yeah, you're on the exactly right. So what we are doing, for example, is so we have new powdery mildew pest disease-resistant materials coming, and what we are doing is in this final round of crosses is to cross these materials with Cabernet, with Pinot Noir, with Chardonnay, to have all these different profiles that are very similar to these materials I just described. Kind of with the idea of doing that, because there is no such a thing as integrating these traits into Cabernet Sauvignon. So, but the moment we make it cross, the whole genomic composition and configuration change something exactly like Cabernet, but with, I don't know, powdery resistant. But we can get something quite similar. The problem also is that grape is very susceptible to inbreeding depression. So what that means is that imagine we cross Cabernet Sauvignon with another one. We have an F1, the progeny. We cannot cross against Cabernet Sauvignon because the plants are going to be very quick and probably are not going to survive. So what we do is the first round we cross it with Pinot Noir, the next one with Zinfandel, and so on. And the final stage, it's what basically impacts the final product. It's Cabernet, for example. So the profile is more similar to Cabernet, but it's not exactly Cabernet. And honestly, I don't know. I think this is something we need to discuss more. What would be the strategy to convince people that this is Cabernet, as you mentioned, and we want to call it Cabernet, but with something different, with an additional trait? I don't know what's going to be the mechanism to do that, to name these new materials like the names of the materials we have now. So
We have another raised hand, Lucia. Go ahead.
Hi, everybody. Can you hear me?
yeah.
Hi. So I have a question that I think might be really unrelated. So I'm very open to being told that that's not an appropriate question. But I did my undergrad at Davis, so that's how I got onto this list. But I now work in coffee. So I live in Guatemala and I work with coffee producers. And one of the biggest, besides, I mean, there's even less genetic diversity in coffee plants. It's insane for how important and how big of an industry it is. But one of the biggest challenges that I see in coffee, besides all of the climate change pressures and prices, is that there's just not enough picking labor. So I was just curious, in California, if anybody could kind of speak to, is that a problem in coffee? In wine, are you seeing that there's less people available to pick? Just any comments on that? And then related to this topic about, breeding, is there any way to have that as a focus? Is there any way to breed for something that would be more easily mechanized, picking more mechanically harvested, something like that? Thank you.
Thank you.
Well, I guess the answer is yes. We have exactly the same problem in the US, right? And I think it's getting worse and worse. I don't have exactly a lot of experience here in California, but yeah, it's definitely a problem. Water and labor is always top 3, top 5 problems in all the meetings I have been. And yeah, so canopy architecture might be a breeding target also. Develop materials that might be more easy to harvest using machines. Probably just management practices, pruning strategies, different trellising systems. Maybe that's enough. Maybe we don't need to attack the problem from the breeding perspective, because canopy architecture is also difficult to work with. It's probably controlled by many genes, and those are the tricky characteristics to work with. I don't know, trellising systems, things like that. Using in bigger rootstocks that promote bigger and reduce bigger. Maybe those are the solutions to have this type of optimal architectures for mechanical harvest.
Okay, I'm going to ask you a question that was sent earlier. It's really somewhere between a comment and a question. And it was, instead of new grape varieties, would you agree that we also need to test old varieties in new regions of the world?
Yeah, I think that's very valid, right? So I was talking with a researcher in Spain. They're looking into more local cultivars they have in Europe. There are hundreds, very well adapted, good wine quality. Why not to use them, right, in a larger scale? But the problem is it's kind of still the same. So it's very hard to do all those evaluations. So where do we want to evaluate them? So we want to have probably common gardens. So we evaluate these plants multiple years, maybe produce wine. I think, yeah, that's we can do that, and yeah, I would say that's a very important thing to do, because we have everything we need already probably available.
I think it's also part of that, the comments that was made about the fact of all these varietals that's already there. The problem is there's actually not as much known about them as what people think. So the more they can get characterized by phenotyping, the better you will know actually whether that variety will be suitable to test in a specific region, to see if it will be suitable for there. You can do that phenotype screening there as well before you go and just plant 20 varieties that you think. You can actually base it on some kind of data.
Yeah, definitely. And then we have to have this force. I don't know if it's coming from the industry, from the sellers, I don't know from where exactly. But even if you have the perfect cultivar, very well characterized, people have to buy it, right?
Yes.
And that's part of the discussion, and I think that's the difficult part, how to sell it.
What are you working on right now?
Well, I'm developing these different type of methodologies to screen plants more efficiently. Not only breeding materials, but also talking about these cultivars that are somewhere that have not been characterized in much detail. We are developing tools to do that, using different like spectral information, robotics, computer vision, things like that are very sensitive techniques that can provide data more efficiently.
Okay. There was also a question about, sorry, Dave, I'm sure it was actually right. There was a question that was sent in regarding the progress in developing powdery mildew resistant varieties. Yeah, so we have this is part of I have done that much in that regard. I just started the position, but I'm just taking over all the materials and the workers that I'm working with. So basically all the credit is for him. So we have materials that are above 90%, 95% vinifera that have multiple resistant genes for powdery mildew and Pests disease. We're going to do more crosses this year to increase the percentage of vinifera and also to have different profiles. So we are crossing these materials with Cabernet and Pinot and others. So we have interesting things coming pretty soon. Okay.
And how much is known about for powdery mildew? How much is known about the mechanism for resistance? Is it just based on the phenotype in the field, or the genetics for resistance understood?
There is a very good amount of research on powdery mildew. So there are multiple species, Asian species and North American species that have resistance to powdery mildew, different genes on different chromosomes, and this is very important because pathogen can overcome resistance very quickly. So it's better to have multiple sources to achieve durable resistance to the pathogen. Many of these genes have already been identified. I know Dario Canto, for example, is doing some work on that regard, how the resistance is regulated. We have molecular markers, so these markers allow us to, so we make a cross, we can run the marker and we will know if that progeny is going to carry the resistance allele, so we can go faster in the case we want to integrate and stack more resistance alleles. So we understand a lot about powdery mildew, not everything, of course, but yeah.
So when you know the gene, you know which genes are important, you can do the, I guess, marker assisted selection. But is it clear like what those genes are doing? Are they changing the surface of the grape or the leaf to make it more resistant to powdery mildew, or is there something different in the function of those?
There's something different, and I am not an expert. I don't have that much experience in that area exactly about what the cellular mechanisms, what the compounds that are being produced that affect the development of the pathogen. But there is a lot of research on that area. We have now this big collaborative project called Viticulture 3. So this is the third iteration. It has been a project the last, I think, 10 years, something like that. And basically the goal of the project was to discover what are all these genes involved in powdery mildew resistance. And at the beginning, we didn't know anything, but now we're in a very good state. So we have about 15 different genes from different species. We know where they are. We know the level of resistance they confer. So they're very strong genes, they're more weaker genes. And researchers and breeders in Cornell, for example, have done a great job in that area. And now in this iteration of the project, the Viticulture 3, that you can do is going to do some gene validation. So knocking out some of these genes to see. So if a resistant if a resistant material has that gene, if we knock it out, supposedly we will lose the function, right? And this is part of the work that it's been done right now.
So I think there's some hands up. This is more an interesting question. A cycle. I can't get down the word, so I'm just going to go ahead. Somebody said, how would you define what you call a new grape variety? Is it something that's lab created with many crossings, or is it a new, an old variety that nobody has used and suddenly it's in favor again, or characterized for the first time? What would you call a new variety?
Yeah, I don't know. It's yeah, it can have different meanings, right? The word new, so new to the world, new to a different country, new to from the breeding perspective, I would say new is something doesn't exist, and we are going to make a cross, and we are going to generate progenies, and we are going to select something that is interesting. That's a new material, right? And, from the breeding,
I think scientifically, it's going to be new. Is going to be something has changed.
Yeah, if there is something growing in Europe and I don't know it, and then somehow we bring it, and then at the end it comes out to be a very important material. Well, that's going to be new to us. I don't know. It's it depends.
That's an interesting one, but yeah, scientifically speaking, I think the definition as new will be that it is different in some way or form. There's something that's actually
exists already. Yeah, exactly.
But as we speak, we usually speak up, even if something it has been around for a while, but you haven't been working on it. You talk about new.
Of course, you think about that, and there are different clones of Pinot Noir or Chardonnay or whatever, and those are presumably genetically different in some way. And how different is different, right? When do you go from a clone to a new variety, or some of the varieties out there are just clones of other things? And I don't know. I mean, I'm assuming some of the relationship between these grapes is known from doing whole genome sequencing and things like that, I guess, right?
Yeah. All the plants, even if they're clones, they change all the time, right? DNA is not a fixed thing. By sexual reproduction, things change considerably, but asexual reproduction, which is supposed to not change anything, changes occur. And that's the case for example, for clones like Pinot or Chardonnay.
I wanted to say a philosophical question, but I just couldn't get on that word. I need more caffeine. So there was a question that was sent in earlier, and that's interesting. They asked, there's a new paper, and I have to say I'm not familiar with this paper about breeding for disease resistance. Please comment on recent research regarding still beans impairing malolactic fermentation. Do you know that?
Well, yeah, I was talking with Dave about it about this. So I'm not familiar with the process in the winery, but what I have seen is that, yeah, so wild species have a higher concentration of these type of compounds, and I think I found a paper that's like 10 times more than vinifera, for example. And there's one paper also that says that, yeah, it inhibits this type of compounds inhibit malolactic fermentation. But something here to remember is that all these materials we are trying to develop, the hybrid grapes, those are very high percentage of vinifera, so above 95%. So we are expecting that the concentration of this type of compounds will be similar to what we found in vinifera. So it shouldn't be a problem. Yeah, if we consider the initial hybrid, right, 50% vinifera, 50% wild species, or purely wild species, non-vinifera species, yeah, this might be a problem. But in this case, that's why it's so important, right, to increase the percentage of vinifera to avoid those type of problems of aromas, things that people don't like about wild non-vinifera species.
So there's a question, I guess kind of a comment, and this gets in the chat that gets back to this idea of, can you breed for to deal with a labor shortage? And this is from Sebastian Traviesa. He writes, for Pinot Noir, we have clones with upright growth, which are easy to mechanize, and clones with dropping growth, which are harder to mechanize. In regards to harvest, certain varieties are easier to pick with automatic harvesters and others are harder. Considering that, using these breeding targets could actually assist with a labor shortage. And I think you kind of mentioned in passing, Luis, that breeding for canopy structure is not the easiest thing, and maybe that's a multi-gene kind of phenotype. Could it be possible to somehow screen for that in a greenhouse?
Yeah, maybe not in the greenhouse. The good thing here is, as Sebastian is mentioning, is so it seems like the trait we are looking for exists, right? And that's a starting point. So we need to combine that phenotype with others, different types of fruit, right? Different, I don't know, white grapes, straight grapes, and so on. And so something, for example, I am doing, which is kind of related with your comment about evaluating this in the greenhouse, is that evaluating plants in the greenhouse is a lot easier because we can have way more. So these are smaller potted plants, and field space is expensive. We have to have people to evaluate the materials in the field, and we have to take care of the plants. So that's expensive, right? So what we're looking, for example, is, at least for drought tolerances, what traits we can measure in small plants in the greenhouse that that inform us about the performance in the field. So that way we can evaluate, let's say, 5,000 plants in the greenhouse and just send to the field the best 20 or 50 that we know are going to perform well. So these are kind of like proxy traits we use to make decisions. So maybe there is something we can measure in the, I'm not sure. Maybe there's something we can measure in the greenhouse. Maybe growth rate or things like that. So we have methods. So we, for example, a robot that has a camera. The camera can move to different places, so we can image thousands of potted plants per day. So maybe there is something associated with architecture and growth rate that we can phenotype with this type of systems.
Yeah, I suppose taking the varieties that are upright or dropping, as Sebastian describes it, growing them in a greenhouse and seeing whether there's a phenotype that could be used for screening early on, because it, like you were saying, to actually wait until canopy develops to choose, you're talking,
And the
years.
problem is always also the environment, right? So maybe one year the environment was allow for more growth. Probably there was more rain or whatever. We have a heat wave, and so we have to have multiple years of data, and we want to have replicated experiments. So that's why it's very expensive to run experiments in the field. But it's very important, right? Because even if I am very precise measuring things in the greenhouse, if those measurements don't mean anything in the field, that's a problem. So I will look into these Pinot Noir clones and try to see.
Yeah, I mean, it'd be interesting to back cross with upright and non-upright clones of Pinot Noir with the same starting material and see how inheritable that.
Yeah, the problem, as I said before, is as soon as you cross things, all these different genes are gonna be rearranged, right?
Yeah.
So that's the problem with the polygenic traits. So that's why these type of traits, from the breeding perspective, need a different approach for breeding.
Or some really good early on phenotype that you can find in a greenhouse that correlates really well with it. Right?
Yeah, exactly.
Yeah, it would be great if only one thing changed at a time.
Well, I suppose that gets you back to the whole CRISPR thing, if there were one gene that controlled each thing, right? But that's
So Luis, you're doing multiple things, but one of the focus areas of your research at the moment is developing those fast throughput phenotyping methods so that you can actually increase the speed of selection, right? Which makes it faster so that you have candidates that you can actually test in the field. So a part of your research is also then to focus on specifically salt and drought tolerant rootstocks, correct? And I assume the thing is that as soon as you have a rootstock that actually give you what you want, have both the traits of salt and drought tolerance, then you screen that salt population to see that they also have disease and nematode resistance, right? Because I look at so many rootstocks that's out there. It's great for this, but not for this. It's great for this, but not for this. And it really makes selection really, really hard because, I mean, almost everybody needs disease resistance. And I know there's areas where this is more problematic than others. But I mean, the holy grail is really to get all those attributes in one rootstock, right?
Yeah, exactly. There are excellent sources for everything. Salt tolerance, bigger, no bigger, drought, and so on, right? The problem is combining them. And I think the other thing we need to think about is that, so if, in one hand, we think about this very unique rootstock that we are trying to develop that combines everything. But maybe that's not exactly what we want to try to do. So we want to stratify, let's say California, for example. So what are the main different regions that have very different soil types and very different conditions, very different production systems. So it's not the same having a 100-acre vineyard in Lodi and a 2-acre vineyard in Napa, for example. So group all of those different scenarios, let's say in 5 different categories. So and then we start to develop into to put on all of those different conditions. I think that's what we need to do. And disease resistance is going to be probably in all of them, right? Because it's a very required characteristic. But maybe we need variation for some other traits, definitely.
So I want to just briefly break here just to say because we have about 5 minutes left, and we'll get back to questions in a second, so you can put your questions in chat. But I want to just pause to thank Karen and Caroline for organizing today's program, and of course Luis for being our guest, and Anita for doing a great job of being co-host as always.
Sure.
I want to remind people that we have a program coming up on May 31st honoring the career of Andy Waterhouse, who Andy retired a year ago, and we're doing a whole day program in honor of him on May 31st today is your last day to register. So it's going to be a really great program. People from industry and academia, former students of Andy's, and other people who have used the knowledge that he's developed over the years. So please go right after this. This is your opportunity to go online and register for that program. In mid-July, we'll be doing an online harvest readiness program. You'll get more information from us about that in the very near future. So with that, and I can see that Caroline put in the link if you want to register for the program honoring Andy Waterhouse. It is right there and easy to click on and chat. So you can do that right now. If there are no other questions in the chat, Anita, do you want to ask the last question?
I don't think there's any more questions from anybody that I have, but I mean, I remember when Louise has that little robot that walks in the vineyard, and I want to sort of ask him, and this may be totally left from what we discussed today, but what is he using that little robot?
Well, we can use that robot to, for example, measure canopy architecture, and I guess it might be important for the discussion we were having. Because all these different traits, like, for example, canopy architecture, these are, some people say, like high dimensional phenotypes. Right? It's very difficult to measure. So you can measure height, you can measure width, but there are many more things that we can see with our eyes, but it's very difficult to measure. And this type of tools allow us to combine like everything we see into multiple or high dimensional phenotypes.
Can you use a robot in the greenhouse?
No, we don't use it in the greenhouse.
Yeah, but it's quite big.
Yeah, exactly. No, yeah, it's for the vineyard, and we have Mason nails. He has also another. I think it's amazing robot. Has more capabilities and things like that. So I think there's a lot of potential in that area for predicting yield, for measuring canopy architecture, and other different traits. So again, everything comes to the, at least from breeding standpoint, the more plants we can evaluate, the better, right? And these tools allow us to do that, increase our screening capabilities.
Yeah. Obtaining a lot of data fast, and then the problem is always sifting through the
analyze.
data. Yeah.
Exactly.
Analyzing the data, yeah.
Yeah, that's just the first step, collecting the data. Then what do we do with the data? That's the problem.
Yes. 1 minute to 3. I'm gonna hand it over to Dave.
All right. Well, again, I want to thank everybody for attending today. It's really great that Luis, less than a year out, has his program up and running, and I think that he would tell you. I know I've heard him say this before, that at the beginning of what we hope is a really long career breeding grapes here, your input is really important. And you can go online and find Luis's email. Please email, engage him, let him know what you think the most important traits are to be looking for. And you can always invite him out to visit you and your vineyard and talk about the problems that you have that you want to see a solution for, even though if it's going to be longer term. So thank you very much, Luis, for being our guest today. And again, thank all of you for attending today's office hours with Dave and Anita. We hope you'll be there for our programs that I just mentioned and our future office hours with Dave and Anita as they come up. So thank you, everyone.
Thank you.
Thank you, everyone. Have a good day.
Thank you.